Mutation. Mutation refers to sudden heritable change in the phenotype of an individual. In the molecular term, mutation is defined as the permanent and relatively rare change in the number or sequence of nucleotides. The term mutation was coined by D. Varese. Classification of mutations. Mutations can be classified in various ways depending upon source, direction, tissue, effects, site, character, and visibility. Agents of mutations. Mutagens. Mutagens refer to the physical or chemical agents which greatly enhance the frequency of mutations. Various radiations and chemicals are used as mutagens. Radiations come under physical mutagens. Physical mutagens. Physical mutagens include various types of radiations, namely X-rays, gamma rays, alpha particles, beta particles, fast and thermal neutrons, and ultraviolet rays. Chemical mutagens. The chemical mutagens can be divided into 4 groups, namely alkylating agents, base analogues, acridine dyes, and others. Mode of action. Substitution. A substitution is a mutation in which there is an exchange between the two bases, that is, a change in a single chemical letter, such as switching AT to AC. Such a substitution could change a codon to one that encodes a different amino acid and cause a change in the protein produced. Example of substitution mutation. Sickle cell anemia is caused by substitution mutation, where in codon GAG mutates to GTG and leads to change. Insertion. Insertions are mutations in which extra base pairs are inserted into a new place in the DNA. The number of base pairs inserted can range from 1 to 1000. Example of insertion mutation. Huntington's disease and the Fragile X syndrome are examples of insertion mutation, wherein trinucleotide repeats are inserted into the DNA sequence, leading to these diseases. Deletion. Deletions are mutations in which a section of DNA is lost or deleted. The number of base pairs deleted can again range from 1 to 1000. Frameshift. Protein coding DNA is divided into codons, which are 3 bases long. Insertions and deletions in these codons can completely change a gene, so its message cannot be decoded correctly. Such mutations are called frameshift mutations. For example, consider the sentence, "The cat ate her rat." Each word represents a codon. If we delete the first letter and read the sentence in the same way, it doesn't make sense. Examples of frameshift mutation: Tay-Sachs disease, cancers of many types, Crohn's disease, cystic fibrosis have been associated with frameshift mutation. Types of mutants. The product of a mutation is known as mutant. It may be a genotype or an individual or a cell or a polypeptide. There are 4 main classes of identifiable mutants, namely morphological, lethal, conditional, and biochemical. Morphological. Morphological mutants refer to the change in form, that is shape, size, and color. Albino spores in Neurospora, curly wings in Drosophila, dwarf peas, short-legged sheep are some examples of morphological mutants. Lethal. In this class, the new allele is recognized by its mortal or lethal effect on the organism. When the mutant allele is lethal, all the individuals carrying such allele will die. But when it is semi-lethal or subvital, some of the individuals will survive. Conditional lethal. Some alleles produce a mutant phenotype under specific environmental conditions. Such mutants are called restrictive mutants. Under other conditions, they produce normal phenotype and are called permissive. Such mutants can be grown under permissive conditions and then be shifted to restrictive conditions for evaluation. Biochemical mutant. Some mutants are identified by the loss of biochemical function of the cell. The cell can assume normal function if the medium is supplemented with appropriate nutrients. For example, adenine auxotropes can be grown only if adenine is supplied, whereas wild type doesn't require adenine supplement.